Hi, welcome back to another session on an important topic, DNA replication. DNA replication is a biological process that occurs in all living organisms and copies their DNA. It is the basis for biological inheritance, and DNA replication is the biological process of producing two identical replicas of DNA from one original DNA molecule. Replication is a process by which genome's DNA is copied in cells. Before a cell divides, it must replicate its entire genome so that each resulting daughter cell ends up with its own complete genome, and this process takes place inside the nucleus as DNA is present inside the nucleus of the cell in case of eukaryotes. In prokaryotes, it occurs in the cytoplasm as there is no nuclear membrane surrounding the genetic material, and the genetic material is present in the cytoplasm, so it occurs in the cytoplasm only. The DNA replication takes place during the synthesis stage of interphase of cell cycle. DNA replication process is said to be semi-conservative, which means that one parent strand is passed down to each daughter strand. And now coming to the major enzymes that are involved in the DNA replication process. These enzymes, they act in coordination so that it helps in the complete process of replication, as each enzyme has a different role to play in the process of replication. The main enzymes that are involved in DNA replication are the DNA helicase, DNA primase, DNA polymerase, and DNA ligase. Let us now see each individual enzyme and its role in the process of replication. First one being the DNA helicase. This helps in the unwinding of the double-stranded DNA molecule by breaking the hydrogen bonds between the complementary base pairs and allowing the DNA strands to separate. For the process of replication to occur, the DNA has to be unwound because it is tightly coiled, and unless it is unwound, the replication process cannot take place. So helicases play a crucial role in replication and repair process of DNA. The second enzyme are the DNA primases. DNA primase is an enzyme which is involved in the replication of replication process and is a type of RNA polymerase. So primase catalyzes the synthesis of short RNA segment, which is called a primer complementary to the short single-stranded DNA template. And the next ones are the important enzymes, which are the DNA polymerases. Eukaryotes contain many DNA polymerases. Of them, alpha, beta, gamma, delta, epsilon, nu are the ones which play a role in replication process of DNA. First, coming to the DNA gamma polymerase, which is located in the mitochondria and is responsible for replication of mitochondrial DNA. As you all know that the replication process of the mitochondrial DNA takes place in the mitochondria itself and has no relation to the replication process that occurs in the nuclear content of the cell. Then DNA polymerase alpha, it binds to the primase enzyme and replicates the first few nucleotides after the RNA primer. Then DNA polymerase delta is the primary DNA polymerase responsible for synthesizing the lagging strand. Similarly, DNA polymerase epsilon is responsible for synthesizing the leading strand. DNA polymerase beta and nu, they help in the repair of the DNA. Then DNA ligases are also referred to as the genetic glue or molecular glue. Glue, as you all know, helps in attaching any of the broken ends. So here the DNA strands which are formed on the lagging strand in the form of short segments, which are called as the Okazaki fragments, they are joined by this molecular or genetic glue, that is DNA ligases. These enzymes, they help in the joining of these Okazaki fragments formed on the lagging strand. Now, coming to the main process or the mechanism of the DNA replication, which is by the semi-conservative method. The steps that are involved in the process are the first one being activation of nucleotides. The four types of nucleotides of DNA, that is DAMP, DGMP, DCMP and DTMP, are present in the nucleoplasm, that is adenine, guanine, cytosine and thymine are present in the nucleoplasm. They are activated by the ATP in the presence of an enzyme phosphorylase. So this results in the formation of deoxyribonucleotide triphosphates, that is DATP, DGTP, DCTP, DTTP by the phosphorylation process in the presence of the enzyme phosphorylase. So the monophosphates are phosphorylated to triphosphates. Now, the second step in the process is point of origin or the initiation point. Now that the DNA is uncoiled, the point of origin of replication process to begin is identified. That replication begins at a point which is the origin of replication represented by O and terminates at a point T. The unit of DNA in which replication occurs is called as the replicon. As you can see in the illustration, the point of replication, you can see it as a bulge there where the forking or for the unwinding of the DNA process has begun. In eukaryotes, there are many such replicons in tandem, whereas in prokaryotes there is only one replicon. In the illustration, you can clearly see many such origin of replications side by side in a row. That is, though you are able to see them as if they are side by side, there are many base pairs apart. And at a point O, endonuclease enzyme temporarily nicks one of the DNA strands on the phosphodiester bond. Then in the step 3, the unwinding of the DNA molecule takes place. So enzyme DNA helicase, it breaks the weak hydrogen bonds at the point of initiation O that was identified in the previous step where the replication is going to begin. That point is the origin of replication. So near that point, the weak hydrogen bonds are broken with the help of the enzyme helicase. The DNA strands start unwinding, which is bidirectional. Each separated strand now acts as a template for replication. The two strands are prevented from recoiling by the enzyme single strand binding proteins, and these proteins remain attached until the synthesis of new strand is completed. As you can see in the illustration, how the unwinding of the two strands of DNA takes place. As both strands are separated, both of them act as templates for the replication process. And once they are uncoiled and separated, the single strand binding proteins attach to these strands and prevent them from coiling back again until the process of replication is completed. Now, the next step is formation of the replication fork. Due to the separation and unwinding of the strands, Y-shaped replication fork is formed. Then the step 5, synthesis of new strands, that is leading and the lagging strands, takes place. For the synthesis of new complementary strand, each template requires a RNA primer. So the primase is the enzyme which helps in this process. So RNA primer attaches to the 3' end of the DNA template strand and attracts complementary nucleotides from the surrounding nucleoplasm. The newly bound consecutive nucleotides are interconnected by phosphodiester bonds, resulting in the formation of a polynucleotide strand. DNA polymerase enzyme catalyzes the synthesis of a new complementary strand which is formed in the 5' to 3' direction. So, in the synthesis of the new strands continues with the formation of a leading and a lagging strand. How it happens, let us see now. The template strand with a free 3' end is called leading template, and the template coding with a free 5' end is called lagging template. As you all know, both the strands of DNA are anti-parallel, that is 3' to 5' will be one strand and the other one will be 5' to 3' end. Since the unwinding has occurred, one end will have the 3' end and the other one will have the 5' end. So the strand, the template strand which is ending with the free 3' end, that is called as the leading template, and the other one which is ending with the free 5' end is called as the lagging template. As both the strands of the parental DNA are anti-parallel, as I've told you, the new strands are always formed in 5' to 3' direction. So the newly synthesized strands develop continuously and discontinuously towards the replicating fork. The continuously synthesized strand is called as the leading strand and is formed towards the replicating fork. The other strand which develops discontinuously is called as the lagging strand as it develops away from the replicating fork. This is clearly shown in the illustration beside the explanation. As you can see, one strand ending with the 3' end, the other template ending with the 5' end. So you can see a continuous strand being synthesized on one of the templates, which is called as the leading strand, and the other one is showing the short fragments being synthesized, which is called as the lagging strand. And the final step is the formation of the daughter DNA. On the lagging template, DNA synthesis takes place in the form of small fragments called as Okazaki fragments. Enzyme DNA ligase joins these Okazaki fragments. DNA alpha polymerase enzyme removes the RNA primers and replaces it by the DNA sequence in eukaryotes. At the end of replication, two daughter DNA molecules are formed. In each daughter DNA, one strand is from the parent and one newly synthesized one. Thus, the replication process is termed as semi-conservative process. That is clearly shown in the illustration here with two different colors, the parental strand in one color and the newly synthesized one in the other color. Since they are carrying one from the parent and one is the newly synthesized one, the daughter DNA molecules, the process is termed as semi-conservative process of DNA replication.